So welcome back again. In whether you decided to go on through the break and join me again for the next recording, or whether you decided to walk away for a while and pick it back up again. Welcome back. Let's take a look at the concept check question very quickly, and then we will move on. So which stage of cell respiration takes place in the cytoplasm? Hopefully you remember that was glycolysis, the universal part of cell respiration, the part that does not require oxygen in order to take place. And what parts take place in the mitochondria? Well, that would be the citric acid cycle and the electron transport process, or as I've all referred to it, as part of a larger complex oxidative process, oxidative phosphorylation. So let's take a look at each of the steps in a little bit more detail. So glycolysis as the first stage is where we start to break down the glucose molecule. In fact, that's what glycolysis means. It means splitting glucose. And so we start with the 6-carbon glucose molecule, and we wind up with 2 pyruvate molecules. Now, math tells you that if you get 2 pyruvate molecules, and you notice it says it splits in half, then a pyruvate molecule, or pyruvic acid, so AKA pyruvate, oh, that's horrible. Let's clean that up. And it has 3 carbons. So the book I know refers to this as pyruvic acid. Typically we refer to it as pyruvate because as an acid, when you dissolve it in water, it gives up its hydrogen ion, becomes pyruvate. So most biochemists and cellular chemists think of this as pyruvate. So you'll hear me refer to it as pyruvate a lot. Because that's how I learned it. So we split the glucose molecule into 2 of these pyruvate molecules. So at the end of the chemical reactions of glycolysis, the glucose molecule has been converted to 2 pyruvate molecules. It's been split in half. So if we're thinking of what goes in chemically, and remember that's one of the things that I want you to focus on, what goes in chemically is a glucose molecule, a 6-carbon sugar. So chemically what comes out is the 2 3-carbon pyruvate molecules. So no real mystery there. I mean, that's pretty straightforward. Along the way, however, as part of the process, we do donate some electrons to reduce NAD plus to NADH. So here's where we start using the term reduction and oxidation. So as part of the glycolysis steps, we do liberate some electrons to make or to reduce NAD plus to NADH. And we actually make 2 NADHs. So we liberate 2 pairs of electrons because it takes 2 electrons to reduce NAD plus to NADH. So we collect 2 pairs of electrons in the form of 2 NADH molecules. And then, having done that reduction step, we generate 4 ATP molecules. 2 ATP molecules at one step, 2 ATP molecules at a second step. Now, the importance of this is that we don't actually get 4 ATP molecules when we, let me erase that and start again. And there's a reason for this. And this is really important because if we are going to think in terms of the energy budget or the ATP output of cell respiration, we need to take this into account. Yes, we get 4 ATP molecules out of glycolysis, but we invested 2 ATP molecules to kickstart glycolysis. So the reality is the overall yield of ATP is only two molecules, the net yield or the overall yield. So we have to take that into account. And then we also generate 2 NADH molecules. So in terms of the overall energy budget, energy in is 2 ATPs, energy out is 4 ATPs for a net yield of 2 plus 2 NADHs. And remember, NADH represents a source of energy because of the presence of those electrons. Ultimately, it is the electrons that are going to give us the big payoff in terms of ATP output. And those electrons in these NADHs produced by glycolysis, they figure into our energy calculations because they do contribute to the overall manufacture of ATP, but that's a little bit further down the road. So if we think in terms of diagramming all of this, we have what is referred to as the energy investment phase, which is the input of the 2 ATPs. At the end of that, we split the glucose molecule into the 2 pyruvates. So no, it's not 2 pyruvates, into 2 intermediates. So you can see that they each have 3 carbons. Then we extract some electrons to reduce NAD plus to NADH, and we do that twice, one for each of these 3 carbon byproducts of the initial split of the glucose. Then, and in the process of doing that, we transfer an extra phosphate in. Then through 2 chemical steps, we transfer both of these phosphates to ADP to make ATP. And in the end, we get pyruvic acid. So we get 2 ATPs here, we get 2 ATPs here. There's our total of 4, but we invested 2 here. So 2 in, 4 out. Net yield to ATP plus 2 NADH, 1 in each of the pathways. And then it's glucose in, two by pyruvate out. So that's the basic summary of glycolysis in terms of the chemical reactions that I really want you to know. Energy in, 2 ATP. Energy out, 4 ATP. Net yield, 2 ATP, plus 2 NADHs. And remember the electrons in those NADHs, while they may be in the wrong place at the moment, they're in the cytoplasm. It is possible to move those electrons, not the NADHs, but the electrons into the mitochondria, where they will contribute to the synthesis of ATP. So those electrons are not lost, they will contribute. Then in terms of the simple chemistry, chemically it's glucose that goes in as the reactant, and what comes out? Pyruvate. Now you'll notice that the overall yield of ATP here is quite small. We're expecting, remember, up to about 32 ATPs net yield or overall yield for cell respiration. So if we've only got 2 at this point, clearly most of the energy of the glucose molecule is yet to be obtained. So where is most of that energy? Well, it's still locked up in the pyruvic acid molecules. So we need to process the pyruvic acid in order to be able to access the remaining energy of the glucose molecule. Because if we do nothing, we're basically throwing away most of the energy of the glucose molecule. And yet there are organisms that do that, as you'll see. So this diagram simply illustrates how the ATP is made. And it's how ATP is made in both glycolysis and the citric acid cycle. And it's called substrate level phosphorylation. Why substrate level? Because it revolves around an enzyme, and you can see the enzyme diagram that in the diagram that transfers the phosphate group from the organic molecule to ADP to form ATP. So that's why it's called substrate level, because it involves an enzyme. Phosphorylation simply refers to the addition of a phosphate group. Anytime you add a phosphate group to something, that's called phosphorylation. So this is substrate level phosphorylation, and this is how the ATP is being made in both glycolysis and in the citric acid cycle, which also produces a small amount of ATP. So now we have an idea of what glycolysis is about. We've got our chemical in, chemical out, energy in, energy out. We know what that's all about. And we've now got a handle on how we make the ATP. But we've got most of the energy of the glucose molecule still tied up in these pyruvate molecules. So the first thing is to get the pyruvate into the mitochondria, because the citric acid cycle, which is going to complete the breakdown of the glucose by completing the breakdown of the pyruvate, occurs in the mitochondria. So the pyruvate moves into the mitochondria by facilitated diffusion. And you might recall that from a previous chapter as a mechanism for getting things across the membrane using a transport protein and a difference in concentration. And the pyruvate can move into the mitochondria very nicely by facilitated diffusion, because there's always a lot of pyruvate in the cytoplasm. That's where glycolysis is making it. And very little in the mitochondria, because that's where it's being consumed by the citric acid cycle. So there is a difference in concentration. All you need is a protein to make it, to allow it to move, and that exists in the mitochondrial membrane. And so it moves into the mitochondria by facilitated diffusion. There, the pyruvate undergoes a grooming process where one of the carbons in the form of a carboxyl group, an acid group, which is a carbon plus two oxygens, is removed. And a carbon plus two oxygens, well, that's carbon dioxide. So now we're seeing the production of some of the carbon dioxide that is given off as the byproduct of cell respiration. The remaining two carbons form what the book calls an acetic acid group, but it's also known as an acetyl or acetyl group. And that needs to be stabilized. And it's stabilized by the addition of this molecule called coenzyme A, or CoA. And so the acetyl group with two carbons is what is going to enter the citric acid cycle. The CoA doesn't. The CoA is simply there to escort the acetyl molecule to the citric acid cycle so that it can be incorporated into the citric acid cycle. So here is the process that's involved. This is the grooming. This is all, so I'll put this here. This is pyruvate. grooming. But notice as part of the process, not only is the carbon dioxide lost, but electrons are transferred to NAD plus and reduce it to NADH. So this is another example of a reduction. And of course, if you've got a reduction going on, an addition of electrons, something else has to be oxidized. So where did those electrons come from? They came from the pyruvate or the pyruvic acid. So this is also known as pyruvate oxidation. So that's what's going on here. So we're picking up another pair of electrons through the loss of that carbon dioxide. And it is the loss of that carbon dioxide that allows us to transfer the two electrons to NAD plus to reduce it to NADH. And those two electrons, they're going to contribute to our energy source. And then you see here the coenzyme A coming in to attach to the acetyl group or acetyl group to stabilize it and escort it to the citric acid cycle. So in the citric acid cycle, the two carbons of the acetyl group attach to a 4-carbon receiving molecule to give rise to a molecule called citric acid, hence the name of the cycle, citric acid cycle. And that's a 6-carbon molecule. As that 6-carbon molecule then passes through the next couple of chemical steps of the cycle, 2 carbon dioxides are released, which is the equivalent of the 2 carbon dioxides that the acetyl group brought in, or the 2 carbons that the acetyl group brought in. So the acetyl group is actually brought into the cycle to replace the carbon dioxides that are given off as the cycle continues. Because for a cycle to continue to function, anything that you lose from it, you have to replace. So in the cycle, we lose 2 carbon dioxides, which means we need to replace those 2 carbons. That's what the acetyl group does to allow the cycle to continue. And with the loss of those 2 carbon dioxides, we've now accounted for the 3 carbons from a pyruvate. Remember, we lost 1 carbon when we groomed the pyruvate, and that was lost as a carbon dioxide. And now we lose another 2 carbons that came from the resulting acetyl group that was produced by this grooming of the pyruvate. So the 1 carbon dioxide that comes from the grooming plus these 2 carbon dioxides from the cycle give us the 3 carbons from pyruvate. And then if we remember that there are 2 pyruvates coming from a glucose molecule, each of which gets groomed and results in the loss of a carbon dioxide. And each then produces its own acetyl group, which in turn enters the citric acid cycle, and then each gives rise to the loss of 2 carbon dioxides. If we keep track of the math, and it gets a little bit difficult to keep track of the math, but if we keep track of the carbon math, we've got 2 carbon dioxides coming free at the grooming stage of the 2 pyruvates, plus we now get 2 times 2 from the 2 acetyl groups coming from the citric acid cycle. That's a grand total of 6 carbon dioxide. There's the 6 carbons coming from the glucose molecule. In other words, the combined action of the pyruvate grooming plus the citric acid cycle gives rise to the 6 carbon dioxides that we exhale every time we breathe out that are the product of the breakdown of the glucose. Something that's important to recognize. It's that grooming step of the pyruvate, plus the citric acid cycle that gives rise to all the carbon dioxide that we exhale. And so it's also the combined action of glycolysis, the grooming step, and the citric acid cycle that leads to the complete chemical breakdown of the glucose from glucose to carbon dioxide. So we've accounted for that side of the chemical equation. We've taken the glucose and we've broken it down to carbon dioxide. And remember, we've already seen the removal of electrons. So this chemical breakdown is also the way in which we extract the electrons. So it also results in the oxidation of the glucose to carbon dioxide. That means that those electrons have been reducing something, and primarily it's been the reduction of NAD plus to NADH. And so that we're going to see as part of the citric acid cycle as well. So let's take a look at a diagram that might help this make a little bit more sense. So here's the basic diagram. So we've got the acetyl group coming in. It binds to the 4-carbon receiving molecule to give us the citric acid. As that then passes through some of the chemicals, the next couple of chemical steps in the citric acid cycle, we get the release of carbon dioxide. So the acetic acid carbon dioxide carbons give rise to the carbon dioxides. The electrons hopefully you can read that over the top. So the 3 NAD pluses that get reduced to NADH, we get 3 pairs of electrons being released as part of the citric acid cycle. Then we have another step that also results in the capturing of electrons, but in this case, it's 2 hydrogens, but remember, an H equals H plus E minus. So when we add hydrogens to FAD to give FADH2, what we have is the equivalent of 2 electrons in the FADH2. And those are electrons that we can also process. So when we think in terms of the citric acid cycle, we've got chemically in the acetic acid, and we've got 2 of those coming in. So we have to, if we're thinking in terms of the overall energy math here and chemical math here, while this shows us what happens for a single pyruvate, so we do need to remember that if we're thinking in terms of a glucose molecule, we have to double down on everything here. So if we were thinking per glucose molecule with double the number of acetyl groups, with double the number of ATPs being produced, we double the number of NADHs being produced, and we double the number of FADH2s being produced. And yes, I didn't mention the ATP, but notice we do get ATP, and this is again substrate level phosphorylation. Getting a little messy there, but it's substrate level phosphorylation that gives rise to this ATP. So we get a small amount of ATP through the action of the citric acid cycle. So we've completed the chemical disassembly of glucose. We've got access to all the energy that we're going to be able to get access to from the glucose molecule. The question now becomes, where is that energy? At the end of glycolysis, it was in the pyruvate molecules, but we've now broken down the pyruvate molecules completely to carbon dioxide. So where is the energy? And at this point, yes, some of the energy is in the small number of ATPs that we got from glycolysis and the small number of ATPs that we get here, but the vast majority of the energy from the glucose molecule is now in the form of the electrons in the NADHs and the FADH2s. So in order to really access that energy and use that energy to make ATP for cellular work, we need to process the electrons. And of course, that means we have to talk about the electron transport process. But before we go there, let's just do a quick concept check. And I've got 3 questions. And this time we're not taking a break. This is just a concept check, not a break in the recording. So what is the chemical product of glycolysis? And in what form does this molecule enter the citric acid cycle? So the chemical product is pyruvate. And it enters the citric acid cycle as the acetyl group. So that's the answer to this question. The chemical product of glycolysis is pyruvate. It enters the citric acid cycle as the acetyl group. What are the processes in your body that result in the CO2 that you exhale? That would be the pyruvate grooming or oxidation, if we're going to give it its proper chemical name, and the citric acid cycle. Oh, that's horrible. Let's rub some of this out. Now, this is a citric acid cycle. So those are the 2 steps that give rise to the CO2 that you exhale. At the end of the citric acid cycle, where is the energy extracted from the glucose located? Some in ATP, but most in the electrons in NADH and NADH2. So that's the energy that we now need to tap into in order to be able to get the energy to make ATP. And that means the electron transport process. So remember, I liken the electron transport process to a cellular staircase. It's a sequence of protein complexes, 3 mainline complexes plus one that sits off to the side, that allows the electrons from NADH and FADH2 to pass down a energy staircase. And as we've seen, going down an energy staircase is the same as going down a playground slide or a slide. And so it's a progressive loss or progressive conversion of potential energy to kinetic energy, which allows us to do work of some form. And in this case, the work that's going to be done is generating a hydrogen ion gradient. The book likens it to damming water behind a dam, building up water behind a dam. And that's not a bad way of doing it. The dam in this case is the inner membrane of the mitochondria. The water is in this case the hydrogen ions that are going to be moved from the matrix of the mitochondrion to the space between the two membranes, what is referred to as the intermembrane space. And that buildup of hydrogen ions is going to be the direct source of energy for driving the synthesis of ATP. And just as water can be behind a dam can be used to drive a turbine to generate electricity, so the hydrogen ions turn cellular turbines, and they really do spin just like a turbine, that drive the synthesis of ATP. They are enzymes called ATP synthase that harvest the spinning energy the spinning that's generated by the flow of the hydrogen ions down through the membrane complex, a protein complex in the membrane. And that spinning turbine is what actually processes ADP and phosphate groups to make ATP. And you have thousands upon thousands of those buried in the inner membrane of every mitochondrion, and every cell in your body has about 30 to 40 mitochondria. So you literally have millions and trillions of these little turbines generating millions of ATP molecules per second. That's literally how much ATP we are making, millions of ATP molecules per second, because we churn through that much on a regular basis. So a simple diagram of what's going on is this diagram. So here you have the NADH bringing in its electrons. Each NADH contributes 2 electrons. And we could also have FADH2 also contributes 2 electrons to the electron transport chain, this cellular staircase that's extracting the energy out of the electrons. In the process of doing that, hydrogen ions from the matrix move to the inner membrane space. Sorry, not inner membrane, intermembrane. That's what I was starting to write here, intermembrane space. And that hydrogen ion gradient is then O grade. I haven't done that for a while. And then the hydrogen ion gradient then is what drives ATP synthesis. So it's not the energy from the electrons that directly drive the ATP synthesis. It's the energy of the electrons creates the hydrogen ion gradient that then provides the energy for driving ATP synthesis. So it's a little bit more complicated than this diagram indicates. The book does have another diagram, and I'm going to show you that diagram in a moment, that ties those together. The electrons, well, ultimately they combine with oxygen and hydrogen ions to form water molecules. So the alternative to doing it this way is to do what NASA does to launch rockets, and when things go wrong there, things tend to go bang and explode. That's what happened to the Challenger. Again, the same amount of energy that was released that caused the Challenger to explode is the same amount of energy that we're tapping into doing it this way. We're just doing it in a more controlled fashion. So there is a huge amount of energy involved here. And we've talked a little bit in previous presentations about how much energy we utilize during the day. So I equated it to about enough energy to raise the temperature of about 2.5 tons of water, about the mass of a small car by 1 degree Celsius. So not surprisingly, make a mistake with doing that and bad things are going to happen. The explosion is truly spectacular when you combine hydrogen and oxygen together. So we are tapping into that same energy. We are just doing it in a very, very controlled fashion. And that means we can access the energy. But even then, the vast majority of the energy is being lost as heat, as we've talked about. So this is the basic nature of the way we tap into that energy from the glucose molecule. We extract the energy from the electrons through the electron transport chain, turn that into a hydrogen ion gradient that spins the turbines as it comes back through the turbines. And those turbines generate the ATP. So this is the diagram that summarizes all of that. So here you can see the components of the electron transport chain. You can see that there are 3 components. So these are the proteins. This is number 1, this is called number 3, and this is called number 4. Number 2 actually sits here and takes the electrons out of FADH2 and enters them into the pathway. But because they enter in a little bit further down, they don't contribute as much energy. And we do take that into account. So the full energy of the electrons from NADH contributes to the buildup of hydrogen ions. The electrons from FADH also contribute, but not as much energy. All of those hydrogen ions then move back through the ATP synthase. It's a facilitated diffusion process. So components of the enzyme act as a facilitator protein complex to allow the hydrogen ions to move through, but that in turn creates the spinning motion of the turbine. And that spinning motion of the turbine is then used to convert ADP and phosphate to ATP. Now, this diffusion process of hydrogen ions is called chemiosmosis. So that's the technical name for the diffusion of the hydrogen ions. And it's the electron transport chain plus chemiosmosis together that is known as oxidative phosphorylation. Now, is it essential for you to know all of these technical terms? No, but as you've been working on your infographic, and of course you have been working on your infographic, you will likely have come across these terms as you do some research outside of the textbook and outside of the PowerPoints. And so because you're encountering those terms, I just want to make sure that you know what those terms are all about. And because the textbook kind of hints at some of these, but never really gets into the details. So there's movement of the electrons through the electron transport chain because its movement of electrons is a sequence of reduction and oxidation reactions. The addition of electrons to NAD plus to give NADH, that's a reduction. The removal of the electrons from glucose and from pyruvate, they're all oxidation reactions. The removal of electrons from NADH as they are added to the electron transport chain, and the removal of electrons from FADH2 as they are added to the electron transport chain, well, they're oxidation reactions. So it stands to reason that the addition of those electrons to the electron transport chain would represent a reduction, and then the movement of the electrons through the electron transport chain from protein to protein is a sequence of reduction and oxidation reactions that result in the loss of energy from those electrons. Ultimately, then, the electrons are combined with oxygen and hydrogen ions to give us water. Not surprisingly, and this is kind of represented here without a whole lot of representation, but you can see the skull and crossbones symbol there. Many poisons, insecticides, and herbicides interfere with the electron transport chain, or ETC to give it its abbreviation. So things like cyanide, carbon monoxide, a common insecticide called rotenone that's also been used for killing rats and mice, so a rodenticide, to give it its correct name, but it's used for killing rats and mice. They all interfere with the passage of electrons through the electron transport chain in various locations. And of course, if you shut down the electron transport chain, eventually you shut down the production of ATP. And there's even an antibiotic that can interfere with the ATP synthase of bacteria. And by shutting down the production of, or the passage of hydrogen ions through the ATP synthase in the bacteria, specifically aerobic bacteria, you kill off the bacteria. So it's, again, a really good way of killing things by shutting down their ability to make ATP, by interfering with aerobic respiration. So in the end, as I mentioned at the beginning, we expect to get around 32 ATPs as a consequence of all of these pieces coming together in the right sequence and applying all of the energy to making ATP. And we can summarize it in the form of this diagram. So in glycolysis, we get 2 ATPs. Remember, we invest 2 ATPs, but we get back 4 ATPs, so we've got a net yield of 2. We get some NADH out of glycolysis as well, 2 of those, and the electrons can be passed into the mitochondria, so they can contribute to the electron transport chain activity and the buildup of the hydrogen ion gradient. When we groom the pyruvic acid to the acetyl-CoA, we again make a couple more NADHs because every carbon dioxide that is lost from a pyruvate allows for the reduction of an NAD plus to NADH. It is, after all, also known as pyruvate oxidation. Once we get the acetyl group into the citric acid cycle, we extract a whole bunch more electrons, and we generate 6 NADHs and 2 FADH2s per glucose molecule. So there we've doubled down on what we saw in the previous diagram, because we're now looking at 2 pyruvic acids. So notice we've got 2 pyruvic acids here, 2 acetyl groups here, which means we've got 2 cycles of the citric acid cycle, which gives us twice that. It also gives us the 2 NADHs here and here, and the 2 ATPs here and here. Now, if all of the electrons from these NADHs and the FADH2s then are processed through the electron transport chain, we generate roughly 28 ATPs. So we get 2 from here, plus 2 from here, plus 2 from, plus 28 from here, which gives us a total of 32. Now, in your investigations, you may have come across the fact that here in the electron transport chain activity, we sometimes refer to 26 to 28, which means 30 to 32 ATPs. 32, let's put it in correctly here. And the reason is because there are 2 different ways for the electrons to get from the cytoplasm into the mitochondria. The more efficient way is to go from an NADH in the cytoplasm to an NADH in the mitochondria. The less efficient way is to go from an NADH in the cytoplasm to an FADH2 in the mitochondria. And so because the FADH2s represent a lower amount of energy, or a small amount of energy, we see this drop off if that's the case. It depends on what type of muscles we're talking about. Is it really important that you know that these differentials? No, but you will have come across them probably in doing your research for the infographic. So I just wanted to point out that that's where that differential comes from. But it's not really important that you understand the nature of where that came from. But in case you were not sure, or you didn't find an answer to that, that's why we have this differential, these flexible numbers. So the other thing that we also need to bear in mind is that while we have been totally focused on burning glucose here as the primary fuel, and cells would prefer to burn glucose, all food molecules, whether it be carbohydrate, whether it be protein, or whether it be fats, lipids, can serve as a fuel. And the reason why is because of how various breakdown products from the different types of food molecules can feed into the cell respiration pathway. So for carbohydrates, it's pretty simple. So long as the carbohydrates can be broken down to simple sugars, which can then be converted into either glucose or fructose. Because in glycolysis, the glucose is actually converted to fructose, and it's the fructose that's actually split to pyruvate. So if you can convert the sugars into either glucose or fructose, then those sugars can enter directly into glycolysis. So long as you can do that, you can pass any of the sugars that are in the food carbohydrates into glycolysis and generate energy that way. When it comes to the proteins, In order to be able to process the proteins, we need to remove amine groups, because the amine groups turn out to be toxic to some of the enzymes in the cell respiration pathway, specifically the citric acid cycle. So those amine groups are removed in the liver primarily, and then the remainder, what's left over of the amine groups, then enters mostly into the citric acid cycle, but some of the amino acids also give acetyl groups and simple sugars that can enter into glycolysis. Primarily 3-carbon sugars that can then process to pyruvate and pass through that way. What happens to those amine groups? Well, they are converted into urea and uric acid and are filtered from the blood via the kidneys and excreted in the urine. This is the reason why, if you eat a lot of protein, you need to drink a lot of water, because water is important for the function of kidneys. And if you're asking your kidneys to do a lot of work because you're eating a lot of protein, then you need to provide them with the necessary water to continue their function. Otherwise, you run the very real risk of kidney damage, which ultimately may mean the shutdown of your kidneys, temporarily or permanently. And if it's permanent, then you'll have to get dialysis and hope that you can get on the transplant list and find someone who can donate a kidney to you. So this is the reason why high protein diets like the Atkins diet, South Beach diet, any of those that emphasize a lot of protein and very little carbohydrate have a very real possibility of leading to kidney damage, apart from any other problem in terms of favoring one type of food molecule over another type of food molecule, which is never a good thing. Remember, everything in balance, not skewing the balance in one direction versus the other. When it comes to fats, the story there in terms of fat composition or the structure of fats gives you a really good idea as to why fats are so energy dense. So number one, remember, fats in the body are triglycerides. So you've got a glycerol molecule, and glycerol is actually an alcohol of the 3-carbon sugar that is produced as a breakdown of glucose in glycolysis. And it's that 3-carbon sugar that ultimately gets processed to pyruvate. So the glycerol can enter very easily into glycolysis. But that's not the real reason why fats are so energy dense. It's the fatty acids themselves. The fatty acids are broken down in the liver by a process called beta oxidation. And beta oxidation produces 2 carbon units that become acetyl groups. And so the average fat or fatty acid in animal fats, like our own fats, may be 20 carbons long, which will yield about 10 acetyl groups, all of which potentially could enter into the mitochondria, or at least be partially introduced into the mitochondria to enter the citric acid cycle. So whereas a glucose molecule produces 2 acetyl groups, a typical fat may produce many, many more acetyl groups, all of which enter the citric acid cycle and participate in the big energy payoff, the electron transport chain and the production of ATP. Remember, that's where the big energy payoff is. So the fats via the acetyl groups that they get broken down into feed directly into that one part of cell respiration that gives the big energy payoff, which is the reason why fats are so energy dense. And I think I've given you the energy breakdown before. But if I haven't, let me give them to you. Now, if I have, well, it never hurts to see it again. So 1 gram of carbs gives you 4 kilocalories. 1 gram of protein also gives you 4 kilocalories. 1 gram of fat gives you 8 kilocalories. So there you can see that fats are twice as energy dense as the equivalent amount of carbohydrate and protein, which explains also why, if you take in an excess of calories, it gets converted to fat. Because you see how all of these foods feed into cell respiration? It turns out that those chemical reactions can either be directly reversed or have a functional reversal in the case of carbohydrates. So you can't really reverse the chemical reactions of glycolysis because there is an irreversible step in glycolysis. But there is a process called gluconeogenesis that allows you to take pyruvate and turn it into glucose. And from there, you can make all sorts of carbohydrates. You can take the acetyl-CoAs and form fatty acids. And of course, you can turn the 3-carbon sugar from glycolysis into glycerol and create fats that way. And you can take the components of the citric acid cycle and acetyl-CoAs and molecules from glycolysis and make amino acids. We do that all the time. We only need to get 9 amino acids in our diet, the 9 essentials. The others we make ourselves using resources from the cell respiration pathway. But if we have an excess of calories, our body is geared to not wasting those calories. We don't throw them away, we store them. And what's the best way to store them? To make fats. So if you take in too much energy from sugar, or if you take in too much energy from proteins, or take in too much energy from fats, your body uses the cell respiration pathway as a source of making fat for storage, because it can store twice as many calories in the same amount of fat. That's the way it goes, unfortunately. Your body will always out-chemistry you because it's better at the chemistry than you are, unfortunately. So this is the last time that we're going to take a break, and we are going to take a physical break here for a moment. Here are our concept checks. So I will see you on the other side of the break to answer these 2 questions. What is the source of the potential energy that drives the ATP synthase that makes ATP? And which of the 3 stages of cell respiration yields the most ATP? I'll see you on the other side of the break.